Good morning. I would like to welcome you all this morning to Pietranera in Sicily, in Italy. I hope you will forgive me if I talk a little more slowly this morning, because I hope that the audience will be a wide audience, not only of English speakers, but also many people in Italy who have the pleasure of listening to the lecture by Dr. Ammar this morning. Today, the students on the higher educational course Industrial Research Experts in the Cereal Sector in southern Italy, part of the project PON 01-01145 Iskocen, have the pleasure of listening to a Lectio Magistralis taken by Dr. Karim Ammar on durum wheat, the new genetic challenge. Dr. Ammar is senior scientist, breeder, and head of the breeding program for durum wheat and triticale, at one of the most important international centres for maize and wheat improvement in the world, CIMIT. Dr. Ammar, with us this morning, began his studies at the National Institute of Agronomy in Tunisia, and then went on to study in Oregon State University, where he took his PhD in genetics. He's world expert in the genetics of durum wheat and has published numerous scientific papers in the area. So today we have the pleasure of listening to his lecture on durum wheat, the new genetic challenge. Dr. Ammar.
Thank you.
Buongiorno a tutti. Good morning, everyone. It's a great pleasure to be here in Sicily for the first time, as far as I'm concerned. It's very important for me to be here and to know this part of the world, which is probably one of the most important and one of the oldest durum producing area in the world. Durum wheat has been here for many centuries, and a lot of the evolution of this crop has taken place here in the island of Sicily. So it's a great pleasure to be here. I would like to really thank the organizing committee, the University of Palermo, and all of my Italian friends who suggested my name to come and give this conference. And it's also a great pleasure to be in an experimental farm. And I think it's a beautiful idea to bring in students to learn about agriculture, to learn about wheat in an experimental farm in the middle of the Sicilian countryside. So today, because of the wider audience, and I would like to welcome the people who are watching us through the streaming session, because of the wider audience, I have prepared, based on some requests from Dr. Giuseppe, I have prepared some slides and to look at some results of our work in breeding for yield potential, drought tolerance, stress tolerance in general, and a few words about quality, which is so important here. But before I do that, I would like to start by giving a small introduction.
Oh, can you use this one?
So before I do that, I would like to start with an introduction on what CIMIT is. For some of you who may not know what CIMIT is and why does a research institution in Mexico, among all the places, is important for wheat improvement in the world. So, okay. This is the general title of my talk.
Obviously, it's going to be a 3-day interaction with you, with the students. And today is going to be more of a summary of the most important part. I will be talking about CIMMYT and what it does globally. I'd like to spend some time talking about what wide adaptation is and is not. That's very critical for us, but also for countries where you have a lot of diversity in the environment, in the production environment. And I think it's a critical concept that everybody who deals with breeding, whether it's wheat or other cereal crops, needs to keep in mind. And what is our breeding strategy in Mexico at CIMMYT to ensure that we have wide adaptation in our genetic material that we distribute worldwide. I'd like to talk about breeding for yield potential and how we can keep this yield potential high even under water limited conditions and a little bit under heat condition as well. And then finally talk about quality. So what is CIMMYT? Really, CIMMYT is historically came out of the effort of a group of people that were sent to Mexico to solve the self-sufficiency problem in the 40s. And that group of people was headed by Norman Borlaug, who started working in Mexico on wheat and started working really to address some very specific issues. He started working first on trying to make the Mexican wheats resistant to diseases, particularly stem rust. That was the main yield-limiting disease at that time. And then when the area where he was working on started getting irrigation water, they needed to get genotypes that were more adapted to more intensive conditions that would not fall. So he used what we call dwarfing genes that make the plant shorter and more responsive to high input and intense agriculture. And his work really, at the beginning, was focused on Mexico, but it took really the world by surprise and really changed wheat around the world. And that's what started the Green Revolution. And that Green Revolution made, or the genotypes that were produced by this Green Revolution, made made Mexico self-sufficient. It also was very instrumental in mitigating the effect of famine in South Asia, in India and Pakistan, and also contributed very, very dramatically in the increase of production and productivity in North Africa, Egypt, parts of Africa, and other areas around the world. So that Green Revolution, Italy had a very similar one with the great Italian wheat scientist of the 40s, 50s, and 60s. And I would really urge the students to really look up the references from Strampelli on to learn about the Italian Green Revolution that was really done independently of what CIMMYT has done. But that had very, very profound effect on transforming the wheat growing conditions in Italy and has contributed very significantly to improving wheat, especially durum wheat, worldwide. So please, students, look it up. There is a very, very big history of Italian wheat research that you need to be aware of. And then by the 70s, thanks to all this work done in Mexico and around the world by Dr. Borlaug, he was given the World Peace Prize, the Nobel Peace Prize, for his for the humanitarian consequences of his work. So this is the history behind CIMMYT's creation, and banking on or capitalizing on the success of this initiative, the world community started thinking of making international research centers that would help developing countries really address their agricultural resource issues. And CIMMYT was one of the centers that were first created in 1966. It stayed in Mexico for historical reasons because the wheat program started there with Dr. Borlaug, but they also added maize, since maize is a crop that is indigenous to Mexico, and so that's how we became the International Center of Maize and Wheat Improvement. Currently, our headquarters are in Mexico, near Mexico City. We have a couple of other stations in Mexico where we do our work, most of our work in wheat. The maize colleagues have another 2 or 3 stations in Mexico, but we are also decentralized all over the world. About 140 researchers in some 14 offices worldwide. Not all of these offices have research projects. Some of them are just contact offices, but we are present everywhere wheat and maize is grown. Our mission currently is primarily the genetic improvement of wheat and maize. We improve these 2 crops, and the outlets, well, our output is what we call international nursery, which are collections of new or improved genetic lines that we distribute around the world. We have some 600 partners that receive our material, including the national programs here in Italy and in Sicily. It's a little bit too small to see, but I'm pretty sure there are at least 2 or 3 blue points corresponding to the institute or the stations that have evaluated CIMMYT material in the past. We are also, and this is not as well known, but we are also involved very, very much in the improvement of crop management technology, not just genetic, but also agronomy and crop management, specifically in the area of conservation agriculture and the area of precision agriculture more recently. And that is very, very important and a very important output of CIMMYT. The other thing that CIMMYT does, it is a curator and a patron for genetic resources for wheat and maize for humanity. We are conserving all the genetic resources that have been collected by us, by others. Our gene bank has more than 140,000 accession of wheat and related species. And we try not only to conserve these genetic resources, but also to promote their use by breeding programs. Okay? That's the important part of our activity. And finally, just to go back just for a few seconds on what Borlaug did. Borlaug did not just put in a bunch of seed in a packet and send it to India or send it to North Africa. He actually trained people to know how to use them, to learn how to breed more of these varieties, and training was critical, was fundamental to the success of the Green Revolution. And we still continue this work of training and capacity building, whether it's for young, new technicians to train them and enhance their capacity, or hosting visiting scientists or students, and so on. So the human factor is still very important in our activity, and that's why I'm here today. Now, how do we work? Breeding is very, very much localized, usually, in a region or in a country. You need to know your environment. So how does a program based in Mexico address so many environments around the world, so many different conditions? It does not seem logical. And in fact, it is almost impossible to understand if you don't know really the program. So we have a breeding program that involves crossing, selection, and evaluation of material in Mexico. And then this material is distributed around the world, either to private companies, to public research institutions, to national programs. They test and select what they like for what is adapted to their environment, and then go ahead and release it. So our difference, or the difference between us and a normal breeding program, is we do not release varieties. We do not go to the end step of actually giving or releasing seed to farmers. This is done by national program. And we work, our first client are the national program or the research institution. Okay. Now, we have had quite a bit of impact, and this graph shows over a couple of decades how national program in developing country, in this case, have used our material. And if you look at bread wheat, about 80% of the varieties released by national program for spring wheat around the world have some kind of CIMT origin, either direct release or progenitor from CIMT that was crossed. And so there is a very, very big dependence from developing country national programs on our germplasm. In durum wheat it's even more. Because durum wheat is a smaller crop, people do not invest too much, except maybe in some areas around the Mediterranean. But usually people do not invest enough for a full-fledged breeding program in durum wheat, and so they rely on us to provide them the germplasm. So that makes us responsible for much of the variety development around the world, and that's a very big responsibility. If you look in terms of spread of our germplasm, you can see here in this graph in red those countries that have more than 40% of their area in wheat in general sown to CIMT germplasm. So it's quite a bit of territory that is used with our germplasm. And again, that's a lot of responsibility, which means that we really have to be aware of what's going on and what are the limiting factors or the new challenges that are occurring everywhere around the world. We need to keep aware of all these new problems that may arise to be able to respond in time. Now, talking about durum wheat specifically, it is important to us who are working now in 2014 to understand and to be aware of the past of our program. Because a breeding program, the material that I'm handling now, that I'm distributing now, that I'm crossing and selecting now, the genes, the gene combination that exists in this material have been put together 20, 30 years ago. So it is important to understand the history of a program when you start working. And really it started in the 60s with the introduction of the dwarfing genes. It was a little bit more difficult to do in durum wheat than it was in red wheat. Dwarfing gene came with some problem of sterility, seed shriveling, but after a few years of breeding we were able to compensate for that and resolve these problems. And some of the first semi-dwarf varieties, like Hori 69, appeared in the late 60s. And then the next decade, really, we put our act together and we started having more success in distributing and widely adapted material like Mexicali and Cocori that were really the basis of the green revolution in durum wheat for many developing countries around the world. Some of these varieties are still grown today. But really where we got our best successes was in the 80s with the super cultivars like Yavaros. Yavaros is the name of the Mexican cultivar, but sister line of these varieties have been released in basically everywhere durum is grown, including in Italy. It's still the #1 variety in North Africa. Tunisia, Morocco still grow more than half of their acreage with varieties from this cross. Spain, until very recently, still grew the Yavaros lines on the majority of their durum acreage. It has been released all the way from Mexico to Australia. So really it has contributed a lot to taking durum wheat to the level of red wheat in terms of acreage and in terms of also increasing yields at national level in many countries. So based on this success in the 90s, we started working on trying to raise the yield potential. And while we were doing that in Mexico, we kind of lost a little bit our view of what was going on around the world. People were saying from North Africa, from Ethiopia, from India, that our material was getting more and more susceptible to diseases. Leaf rust was a problem, stem rust was a very big problem in Ethiopia, Septoria triticide was a problem in Tunisia and North Africa in general, and we were not able to really address this out of Mexico. We did not have those diseases, so we could not breed for resistance to those diseases from our program in Mexico. And at that time, our capacity to work in different countries was limited. So for a decade there, we kind of lost a little bit some of our relevance due to the susceptibility of our material that could not keep up with the evolution of some of the pathogens. And until 2001, that was the year where I started working at CIMMYT. We have witnessed to the appearance of a new leaf rust race that basically came and destroyed all of our program, but gave us the opportunities to start doing breeding for disease resistance, for leaf rust resistance at least. And it took us about 5 years to rebuild the program. And tomorrow, in our second day, I will be talking to the student very much in detail about breeding for disease resistance, and specifically for leaf rust. But it is, for today, just to say that it has been a traumatic event that made us basically rebuild the program around a few sources of resistance. After we did that, we were able to go back and start addressing issues like combining yield potential, drought tolerance, heat tolerance, and looking at other diseases that are more localized in their impact, like septoria and stem rust. So this is where we are right now. It's a continuum, and we are now at a point where we can actually do some preventive breeding, strategic breeding, without having to worry about keeping the program afloat. But a disease can actually destroy the breeding program, even if it's a breeding program that has 40 years of age. Now, wind adaptation. The reason why I am insisting on this concept is because we have been criticized by many, including within the system, that our varieties were varieties that were the same all the way from Mexico to India, and that we are promoting the concept of one variety for all, that our variety, even if they're widely adapted, they do not have sometimes the local adaptation that is necessary to take the best advantage of local conditions. Well, these arguments are probably not true, in the sense that that's not what we are after. We are not after having one variety for for all the countries, because we are aware that one variety in every country puts farmers in a genetic vulnerability condition. That is true if wide adaptation is seen, is perceived like this. So I would like to take some time to really explain and define and clarify what is wide adaptation and why, for us, it is such an important parameter. So let's go back to what the farmers want. And here, this graph is just theoretical data, things that I've done myself. It's not real data, but it's just to illustrate a point. So say you have 10 genotype, evaluating 10 genotype in your yield trials or your regional trial, and you have your check in green over there, and then you have a line that is the best yielding line. That is a line, say you are in a favorable condition, that's the line you want to distribute to the farmer because it's the highest yielding. That's logical. Anybody can see that. You don't need statistics. You don't need to be a hotshot breeder to know that. However, let's say that for any reason, the conditions of your growing environment get a little bit more difficult. Okay? And then all of a sudden, your yellow line over there, genotype number 10, is among the worst ones. It just collapses when conditions get tough. When water gets limited, for example, where nitrogen is not applied properly. When heat becomes really yield limited. And then all of a sudden, the genotype in red is still among the best. Okay? So although it's not the highest yielding under the most favorable condition, it is probably the most stable one if you take into account bad years. Okay? Now the question is, even in favorable conditions, can you still rely, can always rely, that these conditions are always going to be favorable? Let's say, give an example, irrigated wheat. Okay? Irrigated wheat, you can put all the water you need, all the nitrogen you need, at the time you need. But how many regions around the world can rely on abundant supply of water all the time. Mexico, in the Yaqui Valley where we work, near Ciudad Obregón, they used to have 6, 7 irrigation. Now they have 4 to 5 maximum. Water is becoming limiting there. Even in India, where all the wheat, most of the wheat is grown under full irrigation, they're getting less and less water. Why? Because the water is used by the cities, it's used by the industry. It is not replaced or replenished as fast as it's used. The Nile Valley in Egypt, abundant water for many, many decades. Now the water is being more and more scarce. So even in the fully irrigated area, water is not as abundant as before. 30 years from now, we will have to produce the same amount of wheat under irrigation with half the water probably. With global warming, climate change scenarios, water is going to be more and more scarce. And for us, in breeding, our response time is not 1 year to another. It's decades. We need 10, 12 years to get new varieties out to the market. So we really have to think about how the water is going to be abundant when we do our breeding. And in this case, genotype like the yellow one, genotype 10, is probably not the most viable one in the future. But also, can we rely on conditions being similar all the time? No, things are changing. Everywhere, just yesterday I was talking to somebody and telling me that in Sicily, we don't get the cold weather that we used to. Things are changing. Whether it's cycles or whether it's going to be like that forever, we don't know. But we have to be prepared to have more and more changing and more erratic conditions. Now, let's forget about irrigation and let's talk about dry environments, rain-fed environments, where we rely on the rain to grow a crop, whether it's durum wheat or any crop. It's the same scenario. Good rainfall, you have one genotype that emerges as the best, but it collapses in dry year. This is not something we want. This is the kind of genotype, the red one, that people in Sicily have been trying to identify for many, many years. So really, all of a sudden, all of this concept of yield stability, capacity to produce decent yields under very different conditions, that's a positive thing. But that's exactly what wide adaptation is. Wide adaptation is nothing but yield stability over a wide range of conditions. Okay? It is not, I repeat, it is not the struggle or the voluntary will to breed one variety for everything, for all the environments. Okay? So wide adaptation for us is stability and performance in a wide range of environments. The other thing that you may be aware of here in Sicily, because you have a very changing environment, is that the variability within the same region, say this experimental farm, from 1 year to another, is much, much higher than the variability that exists between Mexico and India, for example. So geographic variability is not always more important than temporal variability within the same location. And that's something that needs to be considered. But while we do that, while we're looking at wide adaptation, at the single genotype that has the plasticity to adapt itself to very, very different conditions, we have to ensure that we have in our collection of germ plants the maximum possible genetic variability. That not all the genotype, that all the genotype are plastic, all the genotype are stable, but within the collection that we distribute, they have different genes for diseases, they have different genes for quality. So we provide to the national program a little bit of an insurance policy against emerging diseases or against new market requirements or against new problems of agronomic related problems. So again, we're not in the business of producing the same variety for everybody. We're just in the business of producing varieties that are strongly buffered against environmental effects. All right. Well, this is for wide adaptability. Now, how do we ensure that? This is a similar graph that I showed before. Our breeding program is very similar to any breeding program around the world. Very similar to the breeding program that the Italian colleague have here in Italy, or any private company has. It starts with crossing to generate variability, selection to basically come up with the lines or the plants that we want, and then a verification step, preliminary evaluation step that we do in Mexico. And then once we have that, we distribute it around the world. All these points here in this map are experiment stations, much like this one, that have received at one point or another over the last 20 years or 30 years our material, have tested it. And the originality or the uniqueness of the CIMIC program is that all of these points, or most of them, will return data, information. That information is stored in the database, it's used by us, it is compiled, summarized, and then used again in crossing. For example, if a line has been very well adapted to in India, in Pakistan, and in Iran, and has done very well over there, we see that, we use it to cross it again, because we know it has the wide adaptation that we like to have. So we get data from around the world to feed back in our crossing program, not just from data that we generate ourselves. So that's why I say we have, yes, a centralized program in Mexico, but it is a centralized program with eyes all around the world. It is so important for us, especially for disease resistance. Many diseases do not exist in Mexico, but they exist in Ethiopia, they exist here in the Mediterranean. We get the information and we cross based on that information to get